Good morning, friends. Today I am going to discuss about the complement system. In this video, we will discuss about the function of complement, complement components, complement activation, and the protein which regulate the complement system. Let us see the overview of the complement system. Complement is the major effector of the humoral branch of immune system. As we know, immune system that contains major two branch. One is cell mediated and another is humoral branch. Complement system is involved in humoral branch. That means it is responsible for the activation of humoral branch. Actually, this complement system is invented in 1890s when Jules Bordet at the Pasteur Institute in the Paris. He observed that the antiserum which is isolated from serum against the bacterium Vibrio cholerae, they able to cause the lysis of bacteria. But after hitting this same antiserum, the activity of this bacteriolytic activity of this complement system is destroyed. From this observation, it is concluded that the complement system is heat labile. That means at higher temperature, it destroys its activity. Now, the complement system, which consists of about 30 different proteins. Complement components, they are generally serum inactive. That means it is in inactive form present in the serum, and this inactive form of complements which is activated sequentially by the cascade mechanism. This complement system is also involved complement receptors, which includes cell surface, recognize activated components. It also includes various regulatory proteins which is involved in the complement system. This protein which regulate the activation and inactivation of different types of complements in the complement system. Now let us see. We will discuss the basic function of the complements. In the basic function of complements, as you can see in the figure, it consists of mainly four basic functions: lysis, opsonization, activation of inflammation response, and clearance of immune complex. Now let us discuss the first function, that is called lysis. That means complement has ability to destroy the various foreign cells by the two main pathway. One is antibody dependent pathway and another is antibody independent pathway. That means the complement itself has able to recognize the foreign cells and able to lyse completely. Second function, that is opsonization, or either you can also call it phagocytosis. Opsonization, that means it is somewhat similar like to the phagocytosis. Complement here able to recognize various receptor on the bacteria and form pathogen complement complex. This complex can able to recognize by antigen presenting cell like the phagocytes. And as we know, phagocyte has ability to intake or uptake the whole bacteria by the phagocytosis process. The third function of the complements is the activation of inflammatory response. As already we know what is inflammation. So we are not going to discuss how the inflammation is to be carried out, but complement has ability to activate the inflammatory responses. And the third most important function, that is the clearance of immune complex. That means, as we know, there is a formation of antigen antibody complex which is present in the circulatory system. This immune complexes can be cleared from the system by the function of complements. So it is able to clear the circulating immune complexes by the cell in the liver and the spleen. So these are the basic four functions of the complements. Again, I revised: lysis, opsonization, activation of inflammatory response, clearance of immune complexes. Now let us see the different properties and characteristics of complements, which is possesses. The complement system are mainly synthesized by hepatocytes. As we know, there are different types of complements present in the system, and all these complements are synthesized by the liver hepatocyte. This is the major producing cells. Now, other than this, there are other cells also have ability to produce the complements, like for example, blood monocytes, tissue macrophages, epithelial cells, as well as the gastrointestinal and genitourinary tract. But the main precursor for the synthesis of this complement is generally it is a fraction of immunoglobulins. Now, if you just see the serum globulin fraction in the serum, it is represented about 5%. Most of the complements, they are circulate in the serum, they are functionally inactive form, and this inactive form of complements, which is considered as a proenzyme. It is also believed that the complement is one type of enzymatic, possesses an enzymatic reaction. So either we can call it is a proenzymes or zymogens, which is generally in inactive form. This inactive form of complements is converted into the active form by the proteolytic cleavage. That means one of the inhibitory fragments from the complements is removed, then only the complement becomes active. So there is a cascade enzymatic reaction which ultimately initiate the activation of complement pathway. Now let us see here how many different types of complement which is exist. Generally, complement components are designated by the numbers like C1 up to C9. Sometimes it is also symbol as a factor T, and sometimes it is given as a tribal names, for example, homologous restriction factor. Now, peptide fragments, which is formed by the activation of complements, which is generally denoted by the smaller letters. For example, here I just give one example. Let us assume C4 is one of the complements, and when this C4 complements is hydrolyzed by some enzymes, it will be converted into two fragments. One is C4A and another is C4B. Now generally the smaller fragment, that means its size is smaller. So smaller fragments, they are generally diffused and involved in some signaling pathway. And C4B, which is a larger fragments that remains bound to the microorganisms or the surface where it is attached. So there is one exception in the given the name